Hey, ladies and gents, welcome back. In this segment, we're going to be talking about lipids. Lipids, good old fats and oils. Lipids, what are they made of? Well, let's think back to carbs. Carbs are made of carbon, hydrogen, and oxygen in that 1 to 2 to 1 ratio. So, the way I remember carbs is I think they contain CHO with a lot of O. And that's significant because remember, oxygen, it's pretty electronegative, it draws electrons toward itself. And so, because of that, carbs will interact with water and dissolve. So you think about like sugar, you can sprinkle sugar in water and it dissolves. So because carbs are CHO with a lot of O, they dissolve in water. They're water soluble. Lipids, on the other hand, are CHO with very little O. So since lipids are mostly carbon and hydrogen, they're nonpolar, and so they interact with water differently. They don't dissolve in water. We think oil and water don't mix, right? And that's because lipids, like oils, are mostly carbon and hydrogen. So there's a lot of energy in the bonds that we see there, but they don't behave in water the same way. And that is actually relevant because if lipids weren't immiscible with water, then life wouldn't exist. And we'll talk about that in a little bit. But you can rearrange lipids in all sorts of different ways, and it leads to different functions. So you can sort of see at the bottom of the screen there some of the different ways in which lipids can be constructed. So let's first talk about some of the basic features. Again, they're CHO with very little O. They don't form polymers the way other carbs do, so that's why they don't technically get that macromolecule designation. But what happens is you got a lot of carbon-hydrogen bonds that are non-polar. And again, we'll talk about the structure of that. Now, before we go ahead, I also want to mention for people who maybe haven't taken AP Chem or it's been a while, you see these sort of zigzag shapes. These zigzag shapes, each, it's called a vertex, each sort of point here, or vertex, I believe, is a carbon atom, and it implies its hydrogen buddies. So here, this would be a carbon, and since a carbon needs to have four bonds, it will have two hydrogen partners here. This is another carbon, and it's two hydrogen buddies. Here's a third carbon, and it's two hydrogen buddies. So when you see these points or these zigzags, just recognize that there are carbon atoms there, and they've got hydrogen atoms bonded to them as well. All right, so let's take a look at what are called triglycerides. Triglycerides get the name because they have a glycerol and then three fatty acid tails. So a triglyceride, it does have some oxygen, right? But notice that there's a lot more carbon than oxygen, and the oxygen is on the interior of the molecule. So again, even though they have some oxygen, the oxygen tends to not be reactive, and so it's going to be immiscible in water or won't mix with water. Now, when you see all these carbon hydrogen bonds, there's a lot of energy there. So lipids, like fats and oils, they hold a tremendous amount of energy. So they're great energy storage molecules. So going to this notion of triglycerides, triglycerides have these fatty acid tails. What I'd like to do is look at these fatty acid tails a little more closely to see a difference between fats and oils. So if you look at, this is just looking at the tails, and here they actually filled in the carbons and hydrogens. So rather than having the zigzags, they draw out the carbons and its hydrogen buddies. So this is looking at each individual tail. Here are the three tails. This is zooming in on one individual tail with a saturated fat. Saturated fats are given that designation because they contain all single bonds, no double bonds. And what that means is that it's saturated for hydrogen. Think about what it means for like the soil to be saturated. If the soil is saturated, it can't handle any more water, right? Can't take in any more water. Well, if a fat is saturated, that means it couldn't possibly hold any more hydrogen. And the circumstance in which it can't take any more hydrogen is if it's all single bonds. Okay. Now, notice that the tail is straight. That's significant because since it has straight tails, if you have a bunch of triglycerides together, with all their straight tails, they can pack in pretty tightly. Next time you're in the classroom, if you look on the wall back by the window, we've got saturated and unsaturated lipids. And the saturated lipids are all packed together. And that's significant because when they pack together like that, they tend to form solids at room temperature. So this is what you see in like butter, right? Butter or lard. those are saturated fats. Now, it tends to be how energy is stored in animals. Okay, so again, with all the single bonds, the fats are saturated. That means they have straight tails. They pack in and form solids like fat or butter or lard, things of that nature. Now, please contrast this with unsaturated fats. If they're unsaturated, that means the tails have carbons that possess some double bonds. With these double bonds, could they theoretically hold some more hydrogen? Yeah, absolutely. So, since they could potentially add hydrogen to that tail, it's called an unsaturated fat. Now, this is what we tend to see in oils, and they are oils at room temperature because when these double bonds form, remember, atoms have very characteristic bond angles, right? So when carbons have these double bonds, you have sort of a bent bond angle like that. So the tails are actually bent, and that keeps the molecules spread out. So again, if you look in the back of the room, the unsaturated lipids with the bent tails, they're more spread out. And when they're spread out, then they tend to remain liquid at room temperature. So triglycerides in plants tend to be oils. And again, oils are oils because the molecules are more spread out at room temperature. Let's see here. I'm guessing you've heard of trans fats. So there are saturated fats, which is what we see in animals, where you see the straight chains there that tend to form solids. And then plants have the unsaturated fats that tend to form oils. So what can happen is a process of hydrogenation. So you can add in hydrogens. So this is sort of an interesting image here. Here you see the saturated fat with the straight tail, the unsaturated fat with the bent tail. Here's a trans fat. So what they can do is actually bubble in some hydrogen. And when they do that, it straightens the tails out. Now, that's done because it can make the lipids more shelf stable. So, a lot of cookies will have hydrogenated oils because it can make the cookies more shelf stable, or it's used a lot in baking. But what they found is that saturated fats and trans-saturated fats or trans fats tend to lead to formation of plaques on arteries and are more associated with heart attacks, strokes, and other forms of cardiovascular disease like hardening of the arteries. So lipids are important, right? You need lipids to live. So what I would suggest is try to get your lipids from plant resources rather than animal resources. Animal lipids, animal fats tend to lead to cardiovascular disease, whereas plant-based lipids tend to sort of promote cardiovascular health. All right. When you try to think of the functions of lipids, it's three. Whoops, you can only see two fingers there. Three things to remember. And the way I think of it, I remember MEM. MEM. MEM stands for membranes, energy, and messengers. Okay. Now, the membrane part is the phospholipids and the phospholipid bilayer here. So you see our sort of model for a phospholipid. So all of our membranes are made from lipids. If you didn't have lipids, you wouldn't have cells. You wouldn't have cells, you wouldn't exist. Energy, certainly you think of triglycerides, right, with all those carbon-hydrogen bonds and the energy stored in that. So here you see some fatty tissue in a cell. These are cells called adipocytes. So you can see like a little nucleus in the corner, and it's mostly globules of fat storing those triglycerides for energy. And then finally, you have chemical messengers. Certain types of messengers are steroid hormones or other sort of lipid-based hormones. And these hormones can carry chemical messages all the way into the nucleus of cells, switching genes on or off. So again, when you think about what lipids do for us, just remember MEM, membranes, energy, and messengers. So let's talk about membranes. Phospholipids are a type of lipid. They've got a polar head and then non-polar tails. So what happens is if you take these phospholipids, they will sort of self-organize into a bilayer where the polar heads are on the outside and then those non-polar tails made of carbon and hydrogen are on the inside. Now, if you look, you see the non-polar tails on the inside. You see how they're sort of wriggly like that? Well, that has some very special properties that we'll talk about, or that causes some very special properties of membranes that we'll talk about in, I think, chapter 6. But what phospholipids do is because they have these non-polar tails and polar heads, they arrange in the bilayer And what that bilayer does is it creates an internal environment that has some separation from the external environment. If you didn't have this separation that's created, then you couldn't have the unique chemical reactions that happen inside of cells, and life wouldn't happen. Okay, so again, you think about how like oil and water don't mix, and that seems really pedestrian, but this idea of phospholipids arranging in such a way as to avoid water, it means that you can have an internal environment that's separate, at least partially separate from the external environment, and it allows life to happen. Here is a nice zoom in of individual phospholipid. So you look at the head. Why would the head be polar? Well, look, you got nitrogen and oxygen out the wazoo here, right? So with all this oxygen and nitrogen here, that makes the head polar. If you look at the tail, it's just carbohydrates, right? So those will be nonpolar. And then what do you know? You got a double bond in there. So then that keeps the phospholipids spread apart. So you have some flexibility in your membranes. All right. Here we look at the messenger component of this. You look at a cholesterol. Easy way to spot a cholesterol, it's 4 fused rings of hydrocarbon. So if you look at a cholesterol, is there oxygen here? Yeah, not a lot, right? So 4 fused rings, and it's mostly carbon and hydrogen. Again, each point here is a carbon atom and its associated hydrogen buddies. Now, cholesterols can act in different ways. One cool way is they can actually insert themselves in membranes. So they can actually help stabilize membranes in certain ways. And again, we'll talk about this more in the cell chapter. All right. I think we'll stop there and we can take a quick break. Thanks a lot, folks.